next, we have Dr. Suhasa Kodandaramaiah, and he's going to talk about technologies for understanding the brain. And then, again, keep in mind as you're listening, any questions you might have. All right? And then here it is.
This is very inspiring to me as well, because when Teresa and others contacted me, I had to really dig deep to find these pictures, right? It was kind of going like that. So, about me, I grew up in India. I came to the US when I was 21, right? So for most of my life, most of my childhood, for most of my childhood, I was in India. I included this particular picture here just to illustrate. This is a very funny thing. What we're doing here is undergoing an etiquette class where they taught us how to use a knife and fork because we mostly use spoons, right? And one of the biggest hurdles or barriers that I had to face in my life was actually when I came to the US, right? I had to make a huge cultural shift from relearn everything about life, right? Like use a knife and fork, or learn to live in an entirely new country with a different culture and different expectations and so on. I think that was the biggest challenge, and I was doing that when I had left home, thousands of miles away from my parents and living alone for the first time. And grad school really kicked me in the butt, right? I came here for grad school, and it was a steep learning curve. Anyway, so I grew up in India. For the most part, I was also extremely privileged growing up in India. Like both my parents were doctors, and in fact, looking back, the reason why I'm here is because I was lucky to have been born in that privilege that allowed me to come to the US and pursue higher education and so on, right? So you can see, happy-go-lucky, and then, life kicks you in the butt, right? I got 3 graduate degrees, 2 graduate degrees in the US. I got a master's in mechanical engineering from the University of Michigan. I went to Georgia Tech, which is in Atlanta, to get a PhD in mechanical engineering. And then I did what is called a postdoc, which is typically what people do if they're interested in pursuing a career in academia. This is done at MIT. And then 5 years back, I came to the University of Minnesota, where I'm what is called an assistant professor. Right? So that's my journey from India to the stage here. What do we do? Our lab, we are essentially what I would call a lab that is, we build stuff, right? I'm an engineer. I like to build stuff. I became an engineer because I liked playing with Legos. I liked playing with electronic circuits. And I like to build things with my friends, and that's what I still do. Now I get to work with really smart people who create things to help us better understand how the brain works. In a way, we tend to think of ourselves as, tool makers for science. If you think of astronomy, or you think of everything that we've discovered in space, what is that one fundamental technological leap that happened, right? It was this guy here. Is Galileo Galilei. What did he invent? The telescope, right? So that allowed us to look really far into the sky and discover all these objects and discover everything that we know about how the universe was created, right? And it's still an ongoing discovery. Our lab is trying to invent tools to understand how the brain works. That's what we do in a nutshell. And we believe in what Galileo Galilei said, which is, right now we are able to make some measurements, like Dr. Patel showed us these measurements of how seizures are happening at the surface of the brain. What we don't fully understand is what is causing these seizures, right? For that, we need to zoom into the microscopic level into what each individual cell is potentially doing inside the brain and what is going wrong in a pathological disease like epilepsy. And so we try to invent those tools, right? So we try to invent tools that make measurable what we can't measure right now. I'll give you one example of a scientific as well as a technology project that's going on. These are images of various locations on the university campus. How many of you are here on campus for the first time? Some of you have come here before, right? If I say, go to room 611 in Nils Hassel Mohal, how would you do it? You'll open up your smartphone, right? And then you Google Nils Hassel Mohal, and Google Maps or Apple Maps will tell you, this is Hassel Mohal, this is how you walk. And then once you get to Hassel Mohal, maybe you intuit that it's 611, it's in the 6th floor, you go up, right? You're making use of different strategies to get there, right? When I first came to the university, right, all of this was very new to me, right? This is a sculpture in front of the mechanical engineering department. That's my office. This is Bruiningshall, where I teach a class this semester. This is a funny looking sculpture across the street, and this is Goldie the Gopher in front of Kaufman, right? As I've interacted with this campus over time, I have made a mental map, right? If I see Goldie the Gopher, I know 3 floors up is Campus Club. That's where I have lunch, right? I don't need Google Maps to tell me how to get there, right? And our brains are incredibly good at going to novel environments and learning about them and internalizing them, and very quickly figuring out how to get to where we are. In fact, it's one of those things that is critically important to survive in the wild, right? How does the brain do it? We know we have some clue as to how we do it. Sorry, I should have been speaking on the mic. But we make use of our senses, right? We see things, we smell things. Even if you close your eyes, you're in the Mall of America and you're walking past Cinnabon, you know you're in front of Cinnabon because of that distinctive smell, right? You touch. And so we're making use of these senses, and all these senses are being integrated in the brain, and you're creating maps of the world around you. We sort of know this is happening, we don't know how this is happening. And our lab is trying to answer some of these questions. And to do this, we are inventing different tools. Here's one example of a robot that we have developed that takes a tiny microneedle, which is called a micropipette, and it uses a computer algorithm to guide this microneedle, which has about a little tiny opening in a glass capillary, which is 1 to 2 micrometers wide. And it uses computer algorithms to guide this micropipette to a neuron which is a brain cell inside the brain of a mouse. And when we do this, can we progress a little? While we're doing this, so it uses different algorithms, it's sending pulses of voltage and so on. I won't go into the details, but these algorithms allow us to touch these neurons very delicately. These neurons are about 30 to 40 micrometers, so about a fraction of the width of your hair, right? Very tiny and very delicate. And then once we interface with these neurons, we can record their electrical activity. And we can see how, if this neuron is in the part of your brain called the auditory cortex, your auditory cortex is firing cylinders, all cylinders, as you're listening to me, right? And as you're processing this information. So that's one kind of robotic tool that we develop in our group. So you can record from these cells, right? We also are inventing technologies that allow us to see what's happening at a very large scale, right? For example, we're building robots that can do very fine surgical procedures in mice. So here's a robot that is profiling the surface of the mouse skull and using that information to perform a very precise operation where it very gently and painlessly removes the bone, so we can start looking at the brain. And we can use these kinds of robot to implant skulls that are transparent, right? So we can profile the surface of a mouse skull, use that information to create a digital skull using various computer-aided design tools, and we can replicate these skulls, except now they're transparent, using 3D printers, right? And so here's an example of a replacement skull that we can implant on a mouse, and now we can visualize pretty much the entire top surface of the mouse brain. What can we do with tools like this, right? We can use genetically modified mice that have various kinds of fluorescent proteins that change their fluorescent intensity as the brain is more or less active. And so what you're seeing here is a mouse that's fixed under a microscope, and it's thinking, should I go left, should I go right? And we can start imaging the brain activity in real time. Or we can zoom in, and the mouse is actually walking on a treadmill, and we can start looking at what the individual neurons are doing at a very high resolution, right? We can integrate electronics. You might have all seen these images in sci-fi movies where you have these electronic circuits that are interfacing with the brain. We can do that using transparent and flexible electronics. Okay. And then we can also build really tiny microscopes that you can just attach to the mouse. And now, as the mice are walking around, remember the question I said, how do we learn to navigate the world, right? We can start studying these mice as they learn how to navigate mazes. So here's a maze where we have 3 different locations on a circular maze that the mice have never encountered before. One of these circles is actually a hole. The mice don't like to be out in the open, right? You never see them in the daytime. They're hiding away in corners. And so that's a safe spot. And the first time, the mouse is just randomly looking around, searching, and it eventually finds this goal, which is highlighted in green, and then it very quickly, within 15 minutes, knows the next time we introduce it into this maze, it knows, aha, I need to start looking for these dark spots, and it starts doing that. The next day, it knows exactly where to go, right? Very, very rapid learning. We're using tools like those miniaturized microscopes that I showed to figure out what's happening in the brain while these mice are learning these things. So that's what we do. And that's the team I work with. Thank you. Any questions? Yeah. So the things that we can't do with the human brain, right? And which is why we work with what are called model organisms, like mice. Mice have very similar brain structures. So you have a visual cortex that processes visual information. Mice have a visual cortex, and they work pretty much in the same way. You have what is called a hippocampus, where memories are thought to be stored. Mice have a hippocampus too. And so we use model organisms to ask questions that you would normally not be able to ask in humans, right? But what we discover with these model organisms can translate to what's happening in humans. Yes, Ron. Sure, yeah, that's a great question. Like I said, I grew up in India, right? And both my parents were doctors, but for a long time, it was just assumed that I would follow them into the same profession, right? That's what Indian parents do. They're like, yeah, you are either a doctor or you're an engineer. Uh, but I really like building stuff, right? And that's why I sort of decided to go into mechanical engineering because I thought I'll get all the basic knowledge on how to build stuff. And then, but I was always fascinated with biology. And I would say there are two turning points that happened. One, during my master's, I was working on a project that wasn't going anywhere. And we had this brainstorming session with a neuroscientist who said, you're working on all these little worms, and I won't go into the details, but if you could do this, this is a Nobel Prize, right? We brainstormed, that really sort of sparked, maybe I should be thinking about how we can build tools to understand biology. And then I took a course in molecular biology for engineers that really opened me up into how much in biology we absolutely don't know, right? And that sort of, it was a general transition. And then when I started looking for PhD labs, I wanted to specifically work in a lab that did neural engineering, which was building tools to understand the brain. And so that was my transition. Yes?
You know how you said the hippocampus stores information like memories? Does damage in the hippocampus cause these like Alzheimer's or dementia?
Absolutely. So in fact, the hippocampus, we will have our next speaker who's going to talk in depth about this because she's into Alzheimer's, right? Where, this is hippocampus is one of the first parts of the brain that starts atrophying in various neurological disorders. That's a really good question. And I think I'm out of time.
Thank you for the questions. Thank you. All right. I found the mouse here.